So I'm going to give a talk on the understudy kinome today and the progress that we've made in our work. And I don't want to read this, but the integration, an understudy kinase versus a more well-paired kinase is one that its integration into kinase subnetworks is poorly understood. Its function may be poorly understood in its regulation. And there may be limited kinase reagents that can be used to study these kinases. Our initial list had about 230 kinases, and we pared this down to about 190 to 200 kinases that we would consider by this criterion as understudy. If you look at the RFA that came out from NIH with prioritized understudy kinases, they had 135 in total. 14 of these are actually metabolic kinases and or regulatory subunits of a kinase, not the actual catalytic subunit of a kinase. And 16 are pseudokinases. So those of you not in the field, a pseudokinase has a mutated ATP binding domain, and it may or may not bind ATP, and they usually have mutations where they have very poor or no catalytic activity. So they're actually fairly hard to study, but they're actually, the ones that have been characterized have been shown to be quite important in regulation. And then we use a method, one of the methods we use in my lab, we call multiplexed inhibitor beads. And these are a series of beads that have kinase inhibitors bound to them covalently. And you can use these with lysates from any tissue or cell type that you're interested in. You could do it with yeast or zebrafish. The limitation is simply the kinase inhibitors that you have and their interaction with the active site. These are all ATP competitive inhibitors. And then out of this original 230 kinases that we defined as understudied, we capture routinely 198 of these. So this is about 86%. So if you count the number of pseudokinases that have a non-functional ATP binding site, it's actually greater than this 86%. And we can capture more than 450 kinases in total. If we look at all the cell systems that we've studied, we can be able to capture 90% of the known kinome. So I'm going to give you a little bit of data that we've done with breast cancer, but we've done this in a number of other systems as well. But in a short talk, I have to sort of focus. So down in the bottom of the screen are 6 different inhibitors that are used for these kinds of studies. 2 of them are UNC drugs that were designed here in collaboration with Jan and Stephen Fry and our chemical biology group. And so, for example, UNC 2147A is a chimeric inhibitor that we developed that selectively enhances the enrichment of AGC kinases, of which AKT is one of them, because we weren't binding AKT with their other beads. So we have the ability to make custom beads to enhance the coverage of the kinome. And another example is we just made a PI3 kinase bead where we can capture all of the family members of PI3 kinase family, including different subtypes in terms of alpha, beta, gamma, etc. But in this experiment, if we look at A, we simply took, you can subtype breast cancer into 4 subtypes. Triple negative is basal-like and claudin low. There's a HER2 enriched and on a luminal. And so we took multiple cell lines from these, and here I just have 4 cell lines, 1 from each subtype. And we did MIB-MS with different beads. And what you can show in B is a heat map showing that each bead binds a different set of kinases that we used in this 6-bead experiment, 1 bead per column. And some of the beads bind fairly selectively, but for example, 2147A, which binds AKT, is a needed bead because the other inhibitors don't bind AKT. And you can see in G all the kinases captured, including atypical kinases, and green are the understudy kinases. So we have a really good representation of being able to measure functional kinases using this assay. Now, if we just look at baseline kinome profiling, so we're looking at functional kinases, not with a drug perturbation or some stimulation, but just baseline in cell lines. So we have the claudin low, the basal, the HER2, and the luminal. And you look at B in this kind of general heat map, what you can actually show is that you can subtype breast cancers simply by their baseline kinome profile. And to show you how well this works, HCC 1954, so the top right is our triple negative cells, and the bottom left are luminal cells. And what you can see is that with HCC 1954 and SKBR3, they have properties of both the luminal and the basal triple negative subtype. And HCC 1954s are actually a basal-like HER2 positive. So the vast majority of HER2 positive breast cancers are luminal, but there's a subset of basals that actually amplify HER2. And SKBR3s, even though they're considered luminal by RNA-seq, have properties of basal as well. And so you can see that just the baseline kinome profile by the multiplex inhibitor bead binding defines very nicely these different subtypes. And then you can use the label-free quantitation intensity of peptides for each kinase and generate a heat map like we see on the right that clearly defines the different subtypes, and it clearly defines a basal-like versus a HER2 luminal, for example, a basal-like HER2 amp versus a HER2 luminal, and the other subtypes of breast cancer. So you can actually use PCA space that you would predict from the previous slide and show that these 5 different subtypes of breast cancer are clearly discriminated in PCA space. And if you look at the volcano plot on the right, what you can actually show is that the understudy kinases are strongly represented here in green in the subtyping of the different forms of triple negative as well as luminal and HER2 positive breast cancer, and that there's a discrimination of the understudied kinases that are found in a triple negative bias relative to a luminal bias. So we know that the understudied kinases are differentially expressed and involved in differential regulation of kinome of the kinase signaling networks in the different subtypes of breast cancer. And I apologize for a little bit of the fuzziness of this slide, but if you look at kinome binding versus RNA expression, so you can say, well, is this something that you could just get by RNA-seq? If you look at the kinases detected by MMS in the blue, you can see that we cover a very large percentage of the kinome. And if you look at the expressed kinases, 50 reads or greater by RNA-seq, the expression of the kinome. And then the overlay on the right, the yellow denotes overlap. And you can see that there are kinases that we see by MIB-MS that we don't see at a 50 read cutoff by RNA-seq. And there are kinases that we see by RNA-seq that we don't capture by MIB-MS. And if you look at the relationship between MIB-MS binding and baseline RNA-seq, so we're looking at baseline kinome capture, the baseline expression of functional kinases, there's an extremely poor RNA expression and expression of functional kinases. So kinases would be they would correlate RNA expression. And you can see that there are many kinases in each subtype of breast cancer. So it's basal, claudin low, HER2, luminal. I don't have those labeled there. But you can see that there are many kinases that are below the line and many kinases that are above the line. And we know that there's two regulatory properties that are causing this difference that you would never see by RNA-seq. One is the turnover of these kinases. So with the kinases in particular below the line, where the protein expression is much lower than what you see by predicted. If you use proteasome inhibitor, you normalize many of those. So many of the kinases, both studied and understudied, have a turnover that was previously unrecognized, I think, at this scale, that's greater. So that long non-coding RNAs are sort of a new function for a subset of long non-coding RNAs that we're just getting into in our laboratories, also regulating the functional properties of protein kinases. And we think some of the kinases that are above the line that are very stable are being influenced by long non-coding RNAs. And we can talk about this if we're interested. And then we can take this kind of cell line analysis and actually go into subtypes of tumors in patients. I'm showing you primary tumors of triple negative and HER2 enriched. And what we're looking at here is just in the PCA space, the functional kinome that's expressed in triple negative versus HER2.
I'm sorry, can I just ask a question? For us, there's sometimes a cutoff when you're speaking.
Yeah. Are you? I'm cut off.
Well, there's times where you're towards the end of a sentence you get cut off. So if you wouldn't mind, just start from this slide again. Sorry.
Okay. Can you hear me better now?
We can hear you. Yes, we hear you clearly, and then all of a sudden you get cut off. Maybe it's just our phone.
Okay, I'll try not to do that.
It's happening over here too.
Okay, so what you can see is in A is PCA space, and these are primary human patient tumors, triple negative tumors, as well as HER2 enriched tumors. And so we can do the same kind of analysis with needle biopsy. So we can take a good 14-gauge needle biopsy, and we can look at the functional kinome within these tumors and discriminate the properties of the tumor. And I'll come back with this towards the end of the talk about how we're using this in window trials in patient tumors. And you can see that on the right. So that the understudied kinome plays a significant role in this subtyping. Now then, if we take this cumulative data that we've done with multiple cell lines and multiple mass spec runs, you can easily look at these heat maps of the functional expression of the baseline kinome. You can easily discriminate, as you see in A, triple negative from HER2 luminal tumors. If you do further analysis and look at subtype-specific kinases captured by the multiplexed inhibitor beads, you can actually find that we can subtype tumors very effectively with this list of 50 kinases. And we can subtype not only basal and claudin low, but HER2 enriched luminal and basal-like HER2 amplified. So we can use with RNA, but it gives us a very different measure of function within different subtypes of breast cancer. We've also done this in pancreatic cancer, where you can subtype pancreatic cancer, and we've also done it with non-Hodgkin's lymphoma, where we found subtype lymphoma. So this works very effectively to look at baseline kinome profiles. Now, and we've taken our MIB binding data, the multiplex inhibitor bead binding data, and coupled this with phosphorylation, stimulus-dependent phosphorylation of the different proteins, as well as protein-protein interaction maps that have been characterized that are on the web for this, not work from my laboratory with the interaction maps. And what you can do is you can take the kinases that we data and the protein interaction mapping. And you can do this with, as I have a legend down here, the characterized kinases as whole are the understudied kinases. And you can do this by characterized or understudied distinguishing, which is multiplex inhibitor group that allows us to subtype. And when we do this, we come up with 16 different subnetworks that cluster, and you can distinguish these either using our list of characterized and understudied kinases that we capture, or you can do it with the 50 kinases that we've distinguished that allows us to characterize subtypes within breast cancer. So this begins to define, and if you look at the subnetworks, you see both understudied and studied kinases that are found within each one of the subnetworks. So it really begins to show how understudied kinases in these lists, that include the list with the NIH list as well as our list, that the understudied kinases are integrated in the same subnetworks as I would predict, and I think everyone would predict, as the we're actually being able to see how they begin to integrate into these functional networks. And then we can take this to the level of what my lab, one of the things that my lab's really interested in is drug response. And here to trametinib, which is an FDA-approved MEK inhibitor for melanoma, but characterized in clinical trials for many different tumors. And you can take these subnetworks that you see on the left, and each one of these dots for, so subnetwork 1 or subnetwork 2. Subnetwork 1 is an immune response with an innate immune response in terms of cytokine production. Subnetwork 2 is cell cycle. And what we're actually looking at is log2 ratios of changes of the multiplex inhibitor binding. And you can actually see that that kinases are very, very dynamic in their regulation, and that you can see these changes in both inhibition and also increased binding to the inhibitor beats with drug treatment. This is a 24-hour treatment with trametinib of the different cell lines of the different subtypes. Now, what we also know is that the response of the kinome, both studied and understudied, is different to different inhibitors in the same cell type. So targeting PI3 kinase gives you a different pattern of changes in multiplex inhibitor binding and expression that's versus like MEK inhibition or EGF receptor inhibition, etc. On the right, you can look at these different subnetworks, and you can actually show increases in specific kinases as well as decrease in specific kinases. And the size of the circle is related to the intensity of the number of peptides that we actually capture. And what you can see is both understudied as well as in the green letters as well as well-studied kinases are differentially regulated, both inhibited as well as enhanced in their multiplex and inhibitor binding. Now then, what you can show is that in addition to changes in activation of these kinases, that for the subset of kinases, there's a very strong study kinases. These are in some 159 cells treated for 24 hours with trametinib, and there's a list of kinases that are where their transcription is changed twofold or greater. In the graph, red is understudied, blue are and what you can see is that some of the understudied kinases are induced greater than 30-fold. And what we know is that the integration of the transcriptional regulation in response to targeted kinase inhibitors of the understudied with the studied kinases, and there's a coordinate regulation. And so the kinome is much more dynamic than anyone thought, and this includes the understudied kinome as well as the well-characterized kinome. So there's this integration functionally into these subnetworks, and they're coordinately regulated at a transcriptional level. And then we have a paper that we just, that's in revision for cancer discovery, that I should never be confident, but I'm very confident this paper will get accepted. And basically, we looked at genome-wide induction of enhancer remodeling for adaptive transcriptional regulation by the PTEP-B complex. And this is a major mechanism of drug resistance to targeted inhibitors. And what we know is that the understudied kinases, they're enhancer promoters using chromatin IP. They're regulated dynamically and as dynamically as well-studied and well-characterized kinases, including different oncokinases. And what we really have a snapshot of now when we look at the functional kinome and their integration and their regulation in terms of transcriptional regulation at an epigenomic level, the understudied kinome is tightly integrated into the studied kinome in terms of its regulation. And so there's a different set of understudied kinases transcriptionally regulated, for example, in response to MEK-ERK inhibition versus PI3 kinase AKT inhibition. So on the top is, so these are density peaks that you do when you do chromatin IP. Top graph is an antibody to BRD. We're using antibodies to BRD4, plus, minus and plus trametinib, and JQ1 is an inhibitor of BRD4. And you can see that that blocks the binding, the recruitment of BRD4. And where the arrow is with DDR1, that's the promoter. And you can see with trametinib, there's an enrichment of BRD4 at the promoter. And then left of the promoter is the enhancer for DDR1. We actually showed this as the enhancer by using CRISPR to delete this 17 kb fragment. And you can see this enrichment, and this enrichment of the enhancer region is involved in increasing the transcription of DDR1 in response to trametinib. And you can look now, because we do this in a genome-wide level, you can do this with the understudied kinases that are in our list. And you can show that the enhancer promoter regulation of the understudied kinome is integrated and as robust and dynamic as what you see in this, the understudied kinome. So I think this is really beginning to tell you that even though it may be understudied, that these kinases are integrated into these cases that we've characterized in greater detail in the past. I just want to end by showing some of the work that some people in my laboratory are taking right now. And I'll give you an example of two that we consider understudied kinases, AAK1 and GAC. They have overlapping functions. They're associated with clathrin-coated vesicles, and they're involved in regulating AP2 cargo binding during endocytosis. And there's been an increasing interest in AAK1 and GAC because they're implicated, for example, in viral entry through endosomes. And so, for example, with like hepatitis C and now with Zika virus, the ideas are AAK1 and GAC possibly targets for inhibitors that might inhibit viral entry. So our workflow is summarized. Can do knockout. We've also now got the inactive Cas9 CRISPR working where we can do sort of controlled, modest increased expression of specific kinases. And we couple this, our MIB inhibitor binding, I kind of mentioned this before in terms of the phosphorylation state where we use titanium oxide enrichment of phosphoserine phosphothreonine along with antibody capture of phosphotyrosine peptides. So we use label-free quantitation and we look at multiplex inhibitor bead binding of knockout, of the consequence of knockout of specific genes or overexpression of specific genes in terms of the inhibitor binding profile as well as the phosphorylation networks. And then we analyze kinome dynamics and use this to try to identify kinase signaling subnetworks. So we've knocked out both GAC and AAK1, and we can validate this using what seems like standard CRISPR-Cas targeting now. And phenotypically, when we've done this in a couple of cell lines, but in some 159s, for example, there's no phenotypic consequence that we can easily measure. So there's no change in just baseline growth rate. There's no difference in apoptotic response to an apoptotic stimulus. There's no difference in migration. We haven't done invasion yet. Kinome profiles, and what you can see, for example, is a marked decrease. So what we're looking at is the ratio of inhibitor beat binding of kinases with the knockout relative to wild type. So anything that has a value of 1 means that ratio hasn't changed. Anything with a value less than 1 means there's less binding to the inhibitor beats with the knockout of AAK1 in this case versus the, and an increase, for example, like you see with PGFR beta with the tyrosine kinases, shows that you have an increase in binding. And what you can see is this loss of binding, for example, with EGFR and some of the efferents, and an increase in PGFR beta. And we can do this on a kinome-wide scale, and you can see this change in tyrosine kinases in terms of the multiplex inhibitor beat binding. And we can validate this after we did this because we didn't know exactly what we should look for, and the inhibitor beats directed us toward this, and you can see this by Western blots of EGFR, PGFR beta, and efferent A7. So this directs us in terms of what specific changes are happening with the knockout of AAK1 where we had no other noticeable phenotype at the time to be able to pursue. And then some of our choices of targeting with CRISPR-Cas is being driven by our collaboration with Tim Wilson and Bill Zurcher with the Structural Genomics Consortium. And the Structural Genomics Consortium is interested in making chemical probes or chemical tools that are selective for kinases, specific kinases. And they have a criterion that I won't read to you, but we've been able to work with Tim and Bill, and they have selective probes for GAC and AAK1, and importantly, they have structurally related inactive analogs. And so we can take both a genetic knockout approach as well as a pharmacological approach to study the function of these understudied kinases. And we have chemical probes, not all in my laboratory, but that are being developed by Structural Genomics Consortium for a reasonably significant percentage of the understudied kinases that will integrate with our CRISPR-Cas genetic analysis. So I'm going to stop here, and I won't read these where our current research efforts are and how we're trying to integrate these different approaches, but I'll just talk for a second about the last one and trying to translate this kind of study of the understudied kinome in humans. And so my laboratory is involved in several different clinical trials, 3 of which are what are called clinical window of opportunity trials. And so trial has no real benefit to the patient. It's really for scientific purposes that the patient would volunteer for. And so the patient is, like, for example, diagnosed with 2 other trials we're on with triple negative or HER2 positive breast cancer, and the pathologist takes multiple RNA-seq and NIV-MS proteomics on these pre-treatment. And this patient was scheduled for surgery, and so they go on a different drug regime like trametinib, for example, with triple negative for a week, and then we get the primary tumor that's resected. And we can compare by both RNA-seq and proteomically changes in the kinome, and we can look at both the understudied as well as the well-characterized kinases in response to targeted kinase inhibitors. And in this cancer discovery paper I just briefly talked about, we actually show that the response of the kinome in patient tumors in real time is identical to, or very similar to what we see in cell lines and in PDX models. So we actually feel we've been able to, and now we can really look at the understudied kinome in real time and its integration and functional responses in patients and humans as well as we can in preclinical cell line and animal models. So I'll stop there.